Usually, when you step into a clinic with some kind of physical ailment, there is this comforting expectation of mechanical precision, you know?
Oh, absolutely. We really crave that visual confirmation in medicine.
Right, like you fall, your arm swells up, the physician orders an x-ray, and boom, there it is.
Yeah.
A jagged white line right across the radius.
Exactly. The doctor just points to the illuminated screen and says, well, there's your problem.
It is entirely binary, broken or not broken. You can see it, you can categorize it, and most importantly, you can fix it.
And when a pathogen or an injury is macroscopic like that, it feels manageable. It gives the clinician a very clear, defined enemy to fight.
But the moment you step out of the realm of orthopedics or clear-cut bacterial infections and wade into the world of superficial cutaneous diseases, specifically skin infections, that comforting x-ray machine just completely shatters.
It really does. You are suddenly looking at a diagnostic landscape that is incredibly murky.
Because a rash is just a red patch. A scaly lesion could be, an autoimmune flare-up, an allergic reaction, or a microscopic invasion.
And distinguishing between those visually is nearly impossible sometimes.
So to you, the learner, whether you are a medical professional trying to stay ahead of the curve, a veterinary scientist, or just someone who is insanely curious about the hidden forces shaping our biology, welcome to this deep dive.
And the stack of sources we are synthesizing today is just massive.
It's huge.
Yeah, we are pulling from the latest veterinary medicine guidelines, sweeping global epidemiological treatises, highly granular genomic sequencing reports, and advanced clinical mycology data.
And our mission for this deep dive is to completely deconstruct one of the most misunderstood, highly evolved, and suddenly and terrifyingly rapidly mutating worlds in modern medicine, dermatophytosis.
Which most people casually call ringworm.
Right, which is just a terrible name.
It's the worst name.
Because it has absolutely nothing to do with worms. It is not a parasite. It is a highly specialized predatory fungus.
A fungus with an evolutionary timeline that is genuinely staggering when you look at the fossil record.
That is the part that hooked me immediately. We are dealing with a pathogen that has Mesozoic origins. We aren't talking about, a modern disease that jumped to humans a few thousand years ago during the agricultural revolution.
No, not at all. It's so much older than that.
The sources highlight this specific paleontological discovery, the fossils of a Cretaceous mammal called Spinalestes.
Right, the early mammal fossils.
Yeah, so we are talking about a rat-like creature scurrying through the underbrush like 125 million years ago, while actual dinosaurs like the Spinosaurus were walking the earth.
Which is just a crazy image to hold in your head.
And preserved in the fossilized skin and hair of that early mammal is the exact same type of fungal infection we see today. It was eating the skin of our mammalian ancestors before primates even existed.
And it never stopped. It didn't have to. It merely adapted right alongside mammalian evolution.
It just kept evolving.
Exactly. If you look at the epidemiological data today, up to 20% of the global human population is infected with a dermatophyte at any given time.
Wait, 20%?
Yes. 1 in 5 people walking the planet right now is serving as a host to this ancient lineage of fungus.
That is wild. We dismiss it as this minor itchy annoyance, a little athlete's foot, a bit of jock itch, but biologically, infecting 20% of the dominant species on the planet is an astonishing evolutionary victory.
It is an incredible biological success story.
But here is the critical part, and really the reason we are doing this deep dive today. This ancient fungus is currently undergoing a massive human-driven genetic shift.
A very alarming shift.
We are seeing the global emergence of terrifying superstrains, including a highly aggressive sexually transmitted variant.
Which is completely reshaping how we view the disease.
But before we can even begin to comprehend how a fungus defeats our modern pharmacological weapons, we have to understand the biological machinery of the fungus itself.
And to do that, we have to zoom way out to the kingdom level. The fungal kingdom is vastly different from the plant or animal kingdoms, and it is incredibly dense.
So where do dermatophytes fit into all of that?
They belong to a specific phylum called Ascomycota, which are often referred to as sac fungi, and they fall under the family Arthrodermataceae.
Okay, Ascomycota.
Right. Now, to put their specialization into perspective, mycologists have documented roughly 50,000 distinct fungal species across the globe.
50,000. And those are just the ones we have actually formally categorized, right?
Exactly. The actual number in nature is likely much, much higher. But out of those 50,000 known species, Only about 300 possess the biological machinery necessary to be recognized as pathogenic to humans and animals.
Wow, so the vast majority are completely harmless to us.
Absolutely. Most fungi are perfectly content just decomposing dead trees or living symbiotically with plant roots. They have absolutely no interest in mammalian biology.
It's a massive kingdom, but a very, very exclusive club of killers.
Right, and it gets even more exclusive. Out of those 300 pathogens, less than a dozen species are responsible for causing 90% of all superficial fungal diseases in clinical practice worldwide.
So we are looking at an incredibly specialized elite task force of fungi.
We are. The sources categorize the primary culprits, the ones doing the vast majority of the damage to humans and animals, into three main genera.
The big three of the dermatophyte world.
Exactly. Trichophyton, Microsporum, and Epidermophyton. Those three names carry almost the entire burden of this disease.
But reading through the clinical mycology reports, the way these organisms live and reproduce, it honestly sounds like a science fiction concept.
You're talking about the morphological states.
Yes. This biological dichotomy called the anamorph versus the teleomorph state. I really want to spend some time here because it completely shifts how we view an infection.
It is absolutely fundamental to their survival strategy. Dermatophytes exhibit this complex, dual life cycle with two entirely distinct morphological states. They essentially lead a double life.
Like biological undercover agents.
Essentially, yes. The first state is the anamorph state. This is the pathogenic, asexual phase.
Okay, so this is the form it takes when it's actively infecting something.
Right. It is the form the fungus takes when it is actively invading a host, whether that is a human, a dog, or a cow.
So when I see a red, scaly ring on my arm, I'm looking at the fungus in its anamorph state. It isn't mating. It's just cloning itself and consuming my skin.
Correct. In the anamorph state, the sole biological imperative is aggressive expansion and resource consumption via asexual reproduction.
It just wants to eat and multiply.
Yes. It produces spores that are essentially identical genetic clones of itself. But then we have the teleomorph state.
The secret second life.
Exactly. This is the sexual phase, which mycologists historically refer to as the perfect state. And this state does not happen on the living host.
Where does it happen?
It happens out in the environment, primarily in the soil.
Wait, so they drop off the host, land in the dirt, and literally change their entire biological identity.
They do. When compatible mating strains of the fungus happen to meet in the environmental debris, they can initiate actual sexual reproduction.
Which is a huge evolutionary advantage, right?
Yeah, a massive one. Because sexual reproduction allows for genetic recombination, the shuffling of DNA, to create new, potentially more resilient variations of the fungus.
And when they shift from the asexual parasite to the sexual environmental organism, science actually changes their name, which is completely baffling at first glance.
It is a taxonomic quirk, definitely, but it reflects how radically different the two forms look under a microscope.
Give me an example of that.
Well, let's take Microsporum canis, which is a very common strain that infects cats and dogs.
Okay.
When it is living on a kitten, cloning itself and causing hair loss, we call it Microsporum canis. But if those spores fall into the soil, meet a compatible mating type, and undergo sexual reproduction.
The teleomorph state.
Right. The resulting fruiting body is classified under a completely different name, Arthroderma otae.
It is literally a Jekyll and Hyde scenario.
That's a perfect way to look at it.
Dr. Jekyll is Arthroderma otae, just quietly mixing genes in the garden soil, and Mr. Hyde is Microsporum canis, aggressively tearing through the keratin of a puppy. But what happens if a fungus never goes into the soil? What if it never finds a mate?
That is where the classification gets highly philosophical almost. If a pathogenic dermatophyte has no currently identified sexual state meaning, scientists have searched the dirt and searched the labs, but have only ever observed it in its asexual, parasitic anamorph form.
What do they call it then?
It is historically lumped into a catch-all taxonomic category called the Deuteromycota.
And the Latin translation of that is just incredible. The fungi imperfecti.
The imperfect fungi.
I love that. The fungi imperfecti. It implies that without a sexual life in the soil, they are biologically incomplete. Trapped forever in this cycle of endless, aggressive cloning on human skin.
It is a very poetic way to frame a pathogen.
Which leads us directly into how clinical mycology maps these pathogens based on where they actually evolved to live, because they don't all come from the same environment.
No, they don't. The sources divide them into three incredibly distinct ecological groups.
And this ecological grouping is vital right?
Yeah.
Because it predicts how the human immune system is going to react to the invasion.
Precisely. The first group is the geophilic dermatophytes. The prefix geo refers to the earth.
So, soil fungi.
Right. These are your foundational soil-dwelling fungi, such as Nanitsia gypsy. Their evolutionary niche, their primary job in the ecosystem is to decompose keratinous debris right there in the dirt.
Keratinous debris meaning the physical remnants of animals, like
Mm.
when a bird loses a feather, when a wolf sheds its winter coat, or when a human drops dead skin cells on the forest floor.
Exactly. And keratin is an incredibly tough protein. It doesn't just melt away in the rain.
It's structurally very rigid.
Highly resistant to standard bacterial decay. So the geophilic fungi serve as the specialized cleanup crew. They live in the dirt and they break down those shed materials.
They don't want to attack a living creature. They're perfectly happy just eating the scraps that fall to the ground.
They are detritivores, essentially.
But the second group has moved off the ground, the zoophilic dermatophytes.
Zoo meaning animal. At some point in their evolutionary history, these fungi realized that instead of waiting for the hair and skin to fall to the soil, they could just climb onto the animal and eat it at the source.
Which is a terrifying biological leap.
It's highly efficient. Zoophilic dermatophytes have evolved to adapt to specific animal species, which serve as their primary biological reservoirs.
And the specificity here is what really surprised me. We aren't just talking about a generic animal fungus that attacks everything with fur.
No, they are highly tailored.
The sources detail how Microsporum canis has adapted primarily to domestic cats and dogs. Trichophyton verrucosum has tailored its biology specifically to the skin of cattle.
And there are even more niche adaptations.
Yeah, the one that made me pause while reading the scoping review was Trichophyton mentagrophytes variant Aranetae.
The hedgehog strain.
Yes. This is a strain of fungus that has evolutionarily adapted specifically to the European hedgehog.
It really highlights the sheer adaptive power of the Ascomycota phylum. If there is a keratin-rich environment, even on the prickly back of a wild hedgehog, a fungus will eventually evolve to exploit it.
Nature always finds a way. And that brings us to the third and honestly most relevant group for us, the anthropophilic dermatophytes.
Anthropo meaning human, the human specialists.
So these are the ones that target us specifically.
Yes, these are species like Trichophyton rubrum and Epidumaphyton flocosum. They have abandoned the soil entirely. They have abandoned other animals.
They just want us.
They have evolved to host adapt exclusively to the human body. We are their only reservoir, their only home, and their only food source.
Okay, so we have fungi in the dirt, fungi on hedgehogs and cows, and fungi exclusively on us. But how does the physical invasion actually happen?
The biological mechanics of the invasion.
Exactly. I want to get into the pathophysiology of how a dermatophyte actually eats a human being.
Yeah.
Because when you read the breakdown, it is an incredibly violent microscopic process.
It really is a masterclass in targeted biological degradation. The invasion always begins with the fungal spores, which are technically called arthroconidia.
Arthroconidia.
Right. These spores are microscopic, they are incredibly resilient, and they are constantly floating in the air or resting on surfaces around us.
Just waiting for a target.
The entire process initiates when an arthroconidium makes physical contact with the human stratum corneum.
And the stratum corneum is the absolute outermost layer of the epidermis. Like if you look at your arm right now, everything you were seeing is the stratum corneum.
It's often described as a brick-and-mortar wall in dermatology.
Right, where the bricks are the dead skin cells, the corneocytes, and the mortar is this complex matrix of lipids and fats holding them all tightly together.
That is a very accurate model. Those dead skin cells are fully keratinized, meaning they are packed densely with keratin protein and they are non-viable.
They have no nucleus. They aren't alive.
Exactly. They're just structural armor. For the fungus to survive, the spore must adhere to this surface. And this adherence is a very precarious moment for the pathogen.
Because our skin is designed to keep things out.
It relies heavily on local microtrauma, microscopic scratches, abrasions from tight clothing, or even just the friction from walking.
So a perfectly intact barrier is hard to breach, but a tiny invisible scratch is just an open door for the spore.
Exactly. And the second absolute requirement for adherence is moisture. The spores need a warm, humid microenvironment to activate.
Which perfectly explains the demographic spread. We see this constantly in athletes wearing sweaty synthetic gear, or military personnel marching in combat boots, or people living in high-humidity tropical climates.
Moisture and friction are the biological catalysts.
So what happens after it attaches?
Once the spore is securely attached to a keratinocyte and has the required moisture, it germinates. It wakes up.
Like a seed sprouting.
Similar, yes. It begins to push out a highly organized directional hyphal growth.
Hyphae being those long, branching, root-like tubes that fungi use to explore their environment.
Yes, but they are not just passively growing like a plant root, pushing through soft dirt. The stratum corneum is incredibly tough.
So how do they get through the armor?
To penetrate it, the hyphae secrete an arsenal of specialized extracellular proteolytic enzymes right at their growing tips.
Let's break that down for a second. Proteolytic enzymes. These are proteins designed specifically to dismantle other proteins.
Specifically, they secrete a class of enzymes called keratinases.
Because they want to eat the keratin.
Right. Human keratin is an incredibly robust structural protein. It gets its strength from dense networks of disulfide bonds.
Disulfide bonds, that's sulfur atoms locking the protein chains tightly together, right?
Exactly. It is chemically designed to be highly resistant to breakdown. But the fungal keratinases are engineered by evolution to cleave those exact disulfide bonds.
They act like molecular bolt cutters.
Precisely.
Okay.
They are literally dissolving the structural keratin of the host's skin, hair, and nails into a liquid state.
That is horrifying.
They break the massive protein chains down into small, transportable peptides and basic amino acids.
And then what? It just drinks it.
The fungus then uses specific transporter proteins in its cell membrane to absorb this liquid protein soup. It uses your dismantled skin to fuel the continued growth of its hyphal network.
So it's dissolving your structural armor into a protein smoothie, drinking it, and using the energy to grow longer tubes to dissolve even more armor.
It is a brilliant, highly efficient feedback loop.
It's a terrifying feedback loop, and knowing how it eats keratin dictates exactly where we find it on the human body. Which brings us to the clinical manifestations.
The actual diseases we see in the clinic.
Right, and a phenomenon called zoonotic spillover. But first, let's look at the human mapping. In clinical terminology, any dermatophycosis infection on a human is referred to as a tinea.
And the medical community categorizes these tinnias entirely by their anatomical location.
Let's map this out, because the locations aren't random. They reflect the specific microenvironments the fungus prefers. We start with the most famous one, tinea pedis.
Commonly known as athlete's foot.
Right. And the sources note this is frequently an interdigital infection.
Meaning it occurs primarily in the web spaces between the toes.
Which makes perfect sense based on what we just learned.
If you think about the biological requirements, moisture and microtrauma, the interdigital space of a human foot encased in a modern, occlusive shoe is the absolute perfect incubator.
It traps sweat, it generates friction from the toes rubbing together, and the skin barrier just softens up.
Exactly. Then we have tinea unguium, which doctors often call onychomycosis.
The nail infection.
This is the invasion of the nail plates. And this one is notoriously stubborn because the fungus is digging into a massive concentrated block of hard keratin.
So the nail discolors, thickens, and physically crumbles as the keratinases just hollow it out from the inside.
Right. Moving up the body, we have tinea corporis, which is an infection of the glabrous or non-hairy body surfaces.
The trunk, the arms, the legs.
This is the presentation that produces the classic ringworm visual, an erythematous scaling ring with central clearing.
And I want to clarify that central clearing. The center of the ring looks normal because the fungus is eating its way outward in a circle, right? Like a slow explosion.
That's a great visual. The center is relatively clear because the fungus has already exhausted the local keratin supply there and moved toward the expanding outer border.
It ate everything and moved on.
That's right. Then there is tinea cruris, which infects the groin and inner thighs, commonly referred to as jock itch. Again, an area of high moisture and friction.
What about the head and face?
Tinea capitis is the infection of the scalp and the dense hair shafts. Tinea barbae attacks the coarse hairs of the beard and mustache area. Tinea manuum affects the palms of the hands, and tinea faciae appears on the face.
Now, I want to clarify one thing from the sources because it's a very common point of confusion in dermatology. The texts mention tinea nigra and tinea versicolor, but there's a massive biological distinction here.
It is a critical distinction. Despite sharing the tinea naming convention, tinea versicolor, which causes those pale or dark discolored patches on the chest and back, is not caused by a dermatophyte at all.
Wait, it's not a keratin eater.
No, it is caused by a completely different type of fungus, a yeast called Malassezia.
Okay, so what does Malassezia eat?
Malassezia does not eat keratin. It feeds on the skin's natural oils and lipids. It's a fascinating organism, but biologically distinct from the keratin-eating machinery we are discussing today.
So the true dermatophytes are strictly keratin hunters.
Absolutely.
And while the human-adapted strains like trichophyton rubrum cause a lot of the standard athlete's foot, things get incredibly messy during a zoonotic spillover.
Zoonotic transmission is highly efficient.
This is when an infection leaps across the species barrier, right, from an animal host directly onto a human. The scoping reviews gave us some very specific vectors for this.
It occurs either through direct physical contact like petting an infected animal or indirectly via fomites.
Fomites being inanimate objects or environments contaminated with shed hair or skin scales.
Right. As we touched on earlier with the ecological groups, the occupational hazards here are high. Cattle farmers handling livestock are highly susceptible to contracting T. varicosum.
And on the domestic front, the sources emphasize that children are particularly vulnerable to catching Microsporum canis from newly adopted kittens or puppies.
It's extremely common in pediatric dermatology.
Even individuals working in laboratories or pet stores handling rabbits and guinea pigs are frequently exposed to the Trichophyton mentagrophytes complex.
But the clinical presentation of these animal strains on a human is radically different from the human-to-human strains. And this is where evolutionary biology and immunology really collide.
This is the exact mechanism I want to dig into.
Yeah.
Because I've always wondered, why does a patch of Microsporum canis, a cat fungus, look so much angrier, redder, and more violent on a human arm than a standard human fungus like T. rubrum?
If they both just eat keratin, you'd assume they look the same.
Exactly. Shouldn't they look identical?
You would think so, but the visual difference is entirely dictated by host cell-mediated immunity.
How so?
The anthropophilic fungi, the ones that only infect humans, have spent thousands of years co-evolving intimately with our immune systems. They have undergone selective evolutionary pressure to essentially fly under the radar.
They want to be stealthy roommates. They don't want to alert the immune system's heavy artillery.
Exactly, because if the body mounts a massive defense, the fungus gets eradicated.
So they hide.
They actively modulate their chemical signals to cause only a mild, chronic, low-level inflammation. They suppress local immune responses just enough to stay alive and keep quietly digesting your keratin for months or even years.
That's insidious. But the zoophilic and geophilic fungi, the animal and dirt strains, do not possess this evolutionary stealth.
They are completely alien to the human immune system.
So when a child snuggles a kitten and Microsporum canis spores land on their cheek, the fungus just starts aggressively dissolving tissue without any chemical camouflage.
And the child's immune system instantly recognizes this massive, unmasked foreign invader. It doesn't initiate a slow, measured response.
It panics.
It triggers a catastrophic, highly inflammatory immune cascade. Neutrophils and macrophages flood the tissue. The area becomes acutely erythematous. It gets incredibly hot, swollen, and bright red.
In the sources, they detail how severe this can get, especially on the scalp of a child. It can form something called a carion.
Yes. A carion is a severe, localized, bulgy, boggy inflammatory mass.
It sounds awful.
It looks like a massive pus-filled bacterial abscess, but it is actually just the host's own immune system overreacting wildly to an evolutionarily unfamiliar fungal pathogen.
So the tissue damage isn't just from the fungus eating the hair.
No, much of it is collateral damage from the body's own immune carpet bombing.
Which sounds terrifying for a parent and honestly incredibly confusing for a doctor. Because if you have a massive, weeping, inflamed lesion, or even just a scaly red patch that looks exactly like psoriasis, eczema, or contact dermatitis, how do you know what you're fighting?
You cannot just look at a rash and guess.
You just can't. And this complexity brings us to the diagnostic labyrinth.
Labyrinth is the perfect term for it. The clinical mimics are numerous.
And misdiagnosing a fungal infection as an autoimmune condition like eczema can lead to a physician prescribing steroids, which, as we will discuss later, is a disastrous biological error.
A catastrophic error.
So let's walk through how we actually prove the fungus is there. Let's start with the most traditional, rapid bedside tool, the KOH prep, potassium hydroxide. How does this work?
The KOH prep is elegant in its chemical simplicity. The clinician begins by collecting a physical sample. Using a sterile scalpel blade or the edge of a glass slide, they gently scrape the patient's skin.
But the technique here is critical, right? They don't just scrape anywhere.
Absolutely not. They do not scrape the clear center of the ringworm lesion. They scrape the active, advancing erythematous border.
Because the center is a wasteland. The fungus has already eaten the keratin and moved on.
Exactly. The advancing red border is the front line of the invasion. That's where the highest concentration of active fungal hyphae and spores will be.
So the clinician places those microscopic skin scrapings onto a glass slide. But at this point, you can't see the fungus, right?
No, because it is tangled inside a massive cluster of opaque human skin cells. To solve this, the clinician applies a few drops of a 10 to 20% solution of potassium hydroxide KOH.
And KOH is a highly alkaline caustic chemical. What is it actually doing on the slide?
It acts as a powerful clearing agent. Through a process called saponification and alkaline hydrolysis, the KOH chemically dissolves all of the human material.
It just melts it.
It melts the keratinocytes, the cellular lipids, and the structural proteins of the skin into a liquid.
But if it melts all the structural proteins, why doesn't it melt the fungus? They're both living tissue.
Because of a fundamental divergence in evolutionary architecture.
Can I explain that?
Fungal cell walls are not built from standard proteins or lipids like our cells. They are constructed primarily from chitin.
Chitin.
Right. Chitin is a complex, incredibly durable carbohydrate polymer. It's actually the exact same material that makes up the exoskeletons of insects and crabs. And the highly alkaline KOH cannot dissolve chitin. So as the human tissue melts away into a transparent liquid, the chitinous fungal cell walls remain perfectly intact and structurally rigid.
That is brilliant. It just burns away the human and leaves the crab shell fungus behind.
Essentially, yes.
To speed this chemical melting process up, the sources note clinicians often add a chemical called dimethyl sulfoxide, or DMSO, and they can also add a specialized fluorescent stain called chalcofluor white.
Chalcofluor white is a game changer for diagnostics. It selectively binds directly to the chitinin cellulose in the fungal cell wall.
So it highlights it.
Exactly. When the clinician places the slide under a fluorescent microscope equipped with a UV light, the background human tissue remains dark, but the fungal structures glow with an intense, bright blue or green fluorescence.
And what exactly is the clinician looking for in that glowing blue matrix?
They are searching for the physical morphology of the dermatophyte. Specifically, they want to see hyaline, septate, branching tubular hyphae.
Septate, meaning the long tubes are divided into individual cells by internal cross walls, right?
Right. They look like glowing, branching roots crossing over the ghostly outlines of the dissolved human cells.
That sounds beautiful in a very morbid way.
It's very visually striking. And if the infection is on a hair follicle, they might see tight chains of rounded arthroconidia.
The spores.
Yes. If these spores coat the outside of the hair shaft, it's called an ectothrix infection. If the fungus has invaded and packed the internal core of the hair shaft, it's an endothrix infection.
So on paper, this sounds like an absolutely foolproof test. You dissolve the human, the fungus glows, boom, you have a diagnosis. But the epidemiological statistics in the sources paint a much more concerning picture. This test fails constantly.
It does. Because it is highly dependent on both the physical reality of the sample and the skill of the human looking through the lens. The data gives us specific metrics.
Let's look at those.
The specificity of a KOH prep is rated at 1.0.
Let's define that for the non-statisticians listening. A specificity of 1.0 means there are absolutely zero false positives.
Right.
If the clinician looks through the microscope and physically sees glowing branching hyphae, there is zero doubt. You have a fungal infection. Human skin cannot spontaneously generate chitinous tubes.
Correct. But the sensitivity metric is where the problem lies. The raw mathematical sensitivity of the test is listed at 0.74.
Now, mathematically, a sensitivity of 0.74 means the test correctly identifies the disease 74% of the time in a controlled setting, which would imply a 26% theoretical miss rate.
Right.
But the clinical outline explicitly notes that the sensitivity of 0.74 translates to a practical, real-world 15% false negative rate.
This discrepancy between raw theoretical math and clinical outcomes is fascinating.
Why is it only 15% in the real world?
Well, while the mathematical inverse of 0.74 is 0.26, the specific epidemiological data provided in our sources categorizes the effective clinical false negative outcome rate at 15%. This means that when you account for patient volume, varying severities of lesions, and the clinical threshold for determining a true negative versus an indeterminate slide.
Like the doctor saying, I'm not sure, let's retest.
Exactly. Factoring all that in, 15% of patients who actually possess a pathogenic fungal infection will yield a KOH slide that appears completely clear to the attending physician.
So 15% of infected people are walking out of the clinic being told, good news, it's not a fungus, when it absolutely is. Why is this happening? The sources provide adjusted odds ratios, or AORs, that explain these clinical failures.
An adjusted odds ratio tells us exactly how a specific variable like a patient's behavior impacts the likelihood of an outcome, while mathematically controlling for other confounding factors.
And the data reveals a massive variable: prior antifungal use.
Massive. If a patient has been smearing an over-the-counter antifungal cream on their rash for a week before coming to the clinic, the detection rate plummets. The AOR is 0.19.
An AOR of 0.19 means the odds of finding the fungus drop by over 80%.
Because the over-the-counter cream didn't cure the deep infection, but it did temporarily kill off the active fungal elements right on the surface of the stratum corneum.
Which is exactly where the clinician is scraping.
Exactly. The fungus is still alive deeper in the tissue, but the superficial sample they scraped is sterile.
That's incredibly frustrating for diagnostics.
Conversely, the data shows that the skill of the practitioner is paramount. Having dermatologically trained personnel collect the sample increases the detection rate massively.
What's the AOR for that?
The AOR jumps to 3.03.
Meaning a trained dermatologist is 3 times more likely to find the fungus than a general practitioner, simply because they know exactly how deep to press the scalpel and exactly which millimeter of the red border to scrape.
Medicine is as much an art of technique as it is a science of biology.
So if the KOH slide is blank, but the patient is in agony and the doctor is highly suspicious it's a fungus, what are the alternative diagnostic tools? Let's talk about the Woods lamp.
The Woods lamp is a non-invasive, rapid screening tool. It is essentially a specialized handheld black light that emits ultraviolet light at a very specific wavelength.
Between 320 and 400 nanometers, right?
Yes.
And how does shining a UV light on a scalp help find a fungus?
It exploits a metabolic byproduct of specific fungi. For example, if a child's scalp is infected with Microsporum canis from a kitten, the fungus excretes a chemical called pteridine as it metabolizes the hair.
Pteridine.
Right. When the 320 to 400 nanometer UV photons hit the pteridine molecules, they excite the electrons, causing the infected hairs to fluoresce a brilliant, glowing apple green.
So you can see it instantly in a dark room.
Yes, it's very immediate.
But the sources note a massive blind spot here.
A critical blind spot. The Woods lamp only works on a few specific species, like M. canis.
And what about the others?
The vast majority of trichophyton species, which cause the bulk of adult human ringworm, do not produce those fluorescent metabolites.
So you can shine a UV light on a severe trichophyton rubrum infection all day, and it will remain completely dark.
Exactly. It's not a universal tool.
So if the KOH is negative and the Woods lamp is dark, we have to move to the historical gold standard, the fungal culture. This is where we force the invisible pathogen to grow until we can see it with the naked eye.
Fungal cultures are definitive. The clinician takes those skin scrapings and physically inoculates them onto a nutrient-rich growth medium in a petri dish.
And the sources mention 2 common media.
The 2 most common are Saborod dextrose agar, or SDA, and dermatophyte test medium, commonly called DTM.
The chemistry behind DTM is just brilliant. Can you explain how it provides a visual color shift?
DTM is engineered specifically to isolate dermatophytes from the thousands of harmless environmental bacteria and molds that might be on a patient's skin.
Because skin is covered in microbes.
Right. The agar contains antibacterial agents to suppress normal skin flora, and it contains a specialized chemical pH indicator called phenol red.
So the agar starts off looking like a pale yellow gel.
Yes. Now, dermatophytes have a very specific metabolic preference. Unlike standard environmental molds that prefer to consume carbohydrates and sugars, dermatophytes will selectively metabolize the complex nitrogenous proteins present in the agar.
They are essentially doing to the agar what they do to human keratin. They are breaking down the protein.
Precisely. And as they digest these nitrogenous proteins, they excrete alkaline metabolic byproducts into the surrounding gel.
And that changes the pH.
These alkaline byproducts raise the pH of the agar. When the pH shifts from acidic to highly alkaline, the phenol red indicator undergoes a rapid chemical change, turning the entire pale yellow gel into a bright, unmistakable, glowing red.
It is a phenomenal visual confirmation. A glowing red dish means a keratin-eating pathogen is present.
Yay.
But there's a massive logistical problem with fungal cultures.
Time. Fungi are eukaryotes. Their cellular division is complex and relatively slow compared to bacteria.
How slow?
A culture can take anywhere from 1 to 3, sometimes even 4 weeks, to yield a definitive visual result.
And if you have a massive, itchy, inflamed ring slowly expanding across your chest, you do not want to wait a month to start the correct treatment.
No patient wants to wait that long.
which is why modern clinical mycology is abandoning the petri dish
Mm-hmm.
and leaning heavily into genomics. We're talking about PCR.
Polymerase chain reaction, or PCR, assays have truly revolutionized dermatophyte diagnostics. Instead of waiting for a spore to slowly build a colony, a PCR test searches directly for the underlying genetic blueprint of the fungus.
How does the PCR actually identify it? Because human skin scrapings are full of human DNA, bacterial DNA, viral DNA. How does it isolate just the fungus?
The PCR assay uses specially designed molecular primers that target a very specific, highly conserved region of the fungal genome called the internal transcribed spacer, or ITES region.
The ITES region.
Yes. This ITES segment of ribosomal DNA acts like a highly specific biological barcode.
Okay, so it scans for the barcode.
Exactly. The thermal cycling of the PCR machine denatures the sample DNA. The primers bind only to that specific fungal barcode, and the polymerase enzymes rapidly synthesize millions of copies of that sequence until it reaches a detectable threshold.
And the beauty of PCR is that it doesn't require a living, viable spore. It just needs the chemical presence of the DNA.
That's the key advantage.
So even if the patient used an antifungal cream and killed the surface fungus, resulting in a negative KOH prep and a dead culture, the PCR can still detect the dead fungal DNA.
Exactly. It completely bypasses the variables of viability and human error. It can identify the exact species of the pathogen, whether it's T. rubrum or M. canis, with incredible precision.
And how fast is it?
It reduces the turnaround time from 3 weeks down to just 24 to 48 hours.
Okay, so the molecular barcode is scanned, the machine beeps, and we finally have our definitive diagnosis. We know exactly which dermatophyte is eating the patient. Now we have to destroy it.
We transition from diagnostics to eradication.
Right, diving into pharmacology and the incredibly tedious necessity of environmental decontamination.
The medical arsenal for eradicating a dermatophyte is divided broadly into 2 pharmacological categories: topical therapies and systemic oral therapies.
Right. If you have a mild, highly localized infection, say a small patch of athlete's foot or a single ring on your arm, you can usually rely on topical creams or ointments.
The sources list the heavy hitters for topicals.
Clotrimazole, meconazole, and ketoconazole.
Mm.
These are applied directly to the stratum corneum to wage a surface-level war.
But topicals have strict physical limitations.
Like what?
If the infection is extensive, covering large areas of the body, or if it is refractory and keeps coming back.
Or if it's in the hair or nails.
Most importantly, if it involves the dense, impenetrable keratin of the hair follicles or the nail plates, topical creams simply cannot penetrate deeply enough to reach the leading edge of the hyphae.
You can't just rub cream on a fingernail and expect it to reach the fungal roots buried deep in the nail bed.
Exactly. In those cases, the clinician must deploy systemic oral therapies. The patient swallows a pill, the drug enters the bloodstream, and the body delivers the antifungal agent to the skin from the inside out.
What are the main systemic drugs?
The major systemic drugs listed in the protocols are itraconazole, fluconazole, the older drug grisifulvin, and the historical first-line champion of the last few decades, terbinafine.
I really want to spend significant time on terbinafine, because when you take a pill to kill a fungus, you are literally introducing a poison into your body.
You are.
But human cells and fungal cells are actually quite similar. They are both eukaryotes with complex organelles. So how does terbinafine act as a targeted assassin? How does it kill the fungal cell without melting the human liver or human skin cells?
This requires a deep dive into the biochemical mechanism of action.
Specifically regarding an enzyme called squalene epoxidase.
This is truly a masterclass in targeted molecular pharmacology. To understand how terbinafine kills, you first have to understand how a fungal cell builds its armor.
Okay, let's build the armor.
Fungal cells, unlike human cells, rely on a very specific sterile molecule called ergosterol. Ergosterol is woven into the fungal cell membrane. It maintains the membrane's structural integrity, its fluidity, and its permeability.
It's the load-bearing scaffolding of the fungal cell wall. Without ergosterol, the membrane becomes fragile and leaky.
Right. And human cells don't use ergosterol. We use cholesterol for our cell membranes. That evolutionary difference is the target.
So terbinafine targets the ergosterol.
Terbinafine does not attack the existing ergosterol directly. Instead, it attacks the biochemical assembly line that builds the ergosterol inside the fungus.
It sabotages the factory.
It does. Inside a normal fungal cell, the creation of ergosterol is a multi-step process. Early in this assembly line, a precursor molecule called squalene must be oxidized.
Guane.
Yes. The fungal cell uses a highly specific enzyme called squalene epoxidase, or SQLE, to perform this task. The SQL enzyme grabs the squalene, adds an oxygen atom to it, and converts it into a molecule called squalene epoxide, which eventually becomes lanosterol and ultimately ergosterol.
So the SQLE enzyme is the critical bottleneck in the factory.
Yes, and turbinafine is engineered to perfectly fit right into the active binding site of that fungal SQLE enzyme. It binds tightly and selectively inhibits the enzyme's function.
It acts as an immovable roadblock on the assembly line.
And this molecular roadblock triggers a devastating dual lethal effect on the fungus.
Let's talk about the first effect.
The first effect is straightforward starvation. Because the SQL enzyme is blocked, the assembly line halts. The cell stops producing lanosterol, which means it stops producing ergosterol.
So the cell suffers from catastrophic ergosterol depletion.
Exactly. The cell membrane becomes structurally compromised, functionally porous, and unable to regulate the flow of nutrients.
That alone would eventually kill the fungus. But the second effect of turbinafine is what makes it a fungicidal weapon, meaning it actively murders the cell rather than just stopping its growth.
Because the assembly line is blocked at the squalene phase, all of the raw squalene that the cell continues to produce has nowhere to go.
It just piles up.
It hits the turbinafine roadblock and backs up.
Yep.
This causes a massive, rapid intracellular accumulation of raw squalene inside the fungal cytoplasm.
A toxic spill inside the factory.
An incredibly toxic spill. High concentrations of raw squalene are highly lipophilic and inherently toxic to the delicate internal structures of the fungal cell.
What does the squalene actually do to the inside of the cell?
The accumulating squalene physically disrupts the internal cellular lipid vesicles. It literally destabilizes the cell from the inside out until the internal pressure and lipid disruption cause the fungal cell to lyse it, physically bursts open and dies.
It is a brilliantly efficient biochemical assassination. You take a pill, the turbinafine courses through your blood, deposits into the lower layers of your epidermis, and as the fungus tries to eat your skin, it absorbs the drug, backs up its squalene, and bursts.
It's a highly targeted weapon.
But pharmacology is only half the battle. You can swallow turbinafine for a month, completely clear your skin, and then wake up 3 weeks later covered in ringworm again.
Because we have to talk about the physical environment.
Exactly. The arthoconidia, the spores, are insanely, horrifyingly resilient.
The durability of these spores is one of the greatest challenges in veterinary and clinical management. The sources indicate that dermatophyte arthoconidia can survive dormant in the environment for up to 18 months.
The year and a half. They can sit in the carpet, completely dormant, without food or water, just waiting.
Just waiting for a host.
I can imagine the frustration of a patient or a pet owner hearing this. You spend weeks rubbing clotrimazole on your arm or giving your cat oral medications. The rash finally vanishes, and you think you're done.
Oh.
Why is all this cleaning necessary for a little itchy rash?
Because if you rely solely on pharmacology and ignore the environment, you are virtually guaranteeing a cycle of chronic reinfection. Treating the host without treating the environment fundamentally ignores the primary vector of transmission, fomites.
Fomites are any inanimate objects that can harbor and transmit a pathogen. Hairbrushes, bedsheets, towels, couch cushions, the fibers of a living room rug.
Exactly. When a host is actively infected, they are constantly shedding microscopic, spore-laden skin cells and hairs. A single infected cat can drop millions of viable arthoconidia onto a living room carpet.
Millions?
Yes. If you kill the fungus on the cat's skin with medication, but the cat goes back to sleep on a bed saturated with dormant spores that will survive for 18 months.
The moment the medication leaves the cat's system, those spores reattach to the stratum corneum, germinate, deploy their keratases, and the entire cycle restarts.
It's an endless loop without environmental decontamination.
So what is the actual scientifically validated protocol for decontamination? Because reading the veterinary guidelines, this is not a matter of just running a vacuum and spraying some Febreze.
No, it is a grueling multi-step biological purge.
So step 1.
The clinical and veterinary guidelines mandate a strict, rigorous protocol. Step 1 is absolute mechanical capture.
What does that entail?
Chemical sprays cannot penetrate a thick pile of dog fur or a dense layer of household dust. You must meticulously, repeatedly vacuum and dust every surface to physically capture the shed hair and skin cells that harbor the bulk of the spores. And that vacuum bag must be immediately sealed and removed from the house.
Step 2 involves the textiles.
Step 2 is the hot water laundering of all exposed fabrics: bedding, clothing, towels, pet beds.
Just normal washing?
The water must be very hot, and the mechanical agitation of the washing machine combined with surfactants is necessary to physically dislodge the spores from the fabric weaves.
And finally, step 3, the chemical warfare on hard surfaces. And the chemicals required are highly specific.
You cannot just use a generic, mildly scented kitchen wipe. To breach the chitinous armor of an environmental spore, you must use specific, highly concentrated chemical agents.
What do the guidelines recommend?
The guidelines recommend accelerated hydrogen peroxide diluted at a ratio of 1.178 or a powerful bleach solution diluted between 1.0 and 1.32.
And the most important metric here isn't just the chemical itself, it's the contact time.
This is where almost everyone fails. There is a mandatory wet contact time of 10 full minutes.
10 minutes.
Yes. The surface, the floor, the counter, the grooming clippers must remain physically saturated with the caustic chemical for a continuous 10 minutes.
Because it takes that long to get through the kitten.
It takes that long for the chemical to eat through the thick chitinous cell wall and denature the latent genetic material protected inside the spore. If you wipe the bleach off after 2 minutes because the counter looks clean, you have achieved absolutely nothing.
10 minutes of wet contact. That requires an incredible amount of patience, discipline, and industrial-level cleaning.
It's exhausting.
So if we look at the entire picture up to this point, we have a profound understanding of the taxonomy. We know how the keratinases dissolve our skin. We have PCR tests that can genetically identify the fungus in 24 hours. We have targeted squalene epoxidase inhibitors that burst the fungal cells from the inside out. And we have rigorous leach protocols to sterilize our homes. We have mapped out the perfect defense. We should be winning this war easily.
We absolutely should be. Based on the medical advances of the late 20th century, superficial dermatophytosis should be an easily managed, highly localized nuisance.
But we aren't winning.
We are losing ground rapidly.
In fact, the epidemiological treatises in our sources indicate we are standing on the precipice of a massive global failure in dermatological medicine. And that brings us to the final and, frankly, the most alarming section of this deep dive. The antimicrobial resistance crisis, the emergence of the superstrains.
What is unfolding right now in clinical mycology is a fascinating yet terrifying real-time demonstration of how rapidly the evolutionary arms race can escalate when human behavior forces a pathogen's hand.
Let's introduce the main antagonist of this new era, Trichophyton indotini.
Trichophyton indotini is a pathogen that caught the global medical community completely off guard.
Where did it come from?
Originally, when clinicians first started culturing it, it was classified as just another minor variant of an existing species. They cataloged it as Trichophyton mentagrophytes genotype 8.
Genotype 8, identifying it through the ITS ribosomal DNA sequencing we discussed earlier.
Right, but its behavior in the real world was so radically different, and its genetic divergence became so extreme that taxonomists had to rewrite the books. They elevated it and declared it an entirely new, highly virulent, independent species.
Let's talk about what this superstrain actually looks like on a human being, because the clinical descriptions in the sources sound more like a horror movie than a standard case of ringworm.
The clinical presentation is explosive. Standard ringworm is usually a small, localized, manageable ring. T. indotini causes extensive, highly inflammatory, and intensely pruritic plaques.
Pruritic, meaning the itching is so severe it causes significant psychological distress and sleep deprivation for the patient.
Yes.
And these aren't small rings. The sources describe these angry, scaly plaques converging and merging over massive areas of the human body. They cover the entire trunk, sweeping across the abdomen, deeply infiltrating the groin, spreading across the gluteal region, and even ascending up to the face.
And what makes it diagnostically unique and quite strange from an evolutionary standpoint is its anatomical preference.
It exhibits the feet.
Exactly. It aggressively consumes the torso and the groin, but it notably, almost entirely, spares the feet and the nails.
Which is completely counterintuitive because, as we established earlier, the moist, secluded space of the foot is usually the absolute favorite environment for a dermatophyte. But this superstrain ignores the feet and goes straight for the core of the body. Where did this come from and how fast is it moving?
The epidemiological tracing indicates it began as a massive, sweeping epidemic across India and the surrounding South Asian subcontinent. Clinics were suddenly overwhelmed by patients covered in these massive, intractable plaques.
But it didn't stay there.
No. Due to modern global travel and constant human interaction, it did not stay regional. It has breached containment and is now spreading rapidly worldwide. Cases are surging in clinics across Europe, North America, and globally.
But why is it a superstrain? A fungus can be aggressive, but if we have turbinafine to burst its cells, we should still be able to kill it.
Though we can't.
We can't. Let's do a massive molecular deep dive into the exact genetics of its resistance, because this is where the biochemistry of the squalene epoxidase enzyme becomes the center of the story again.
This is the crux of the crisis. The genomic analysis of T indotiny reveals exactly how it defeats our most powerful systemic drug. It all comes down to highly specific, singular point mutations within the fungal DNA that encodes for the sequel EAD.
A point mutation meaning a tiny microscopic typo, a single change in the millions of letters of the DNA sequence.
Yes. The sources detail a specific dominant mutation that has emerged, the C.1179AT substitution.
Okay, let's break that scientific shorthand down for you listening. What does C.1179AT actually mean on a molecular level?
It refers to the exact location of the genetic typo. It means that at nucleotide position 1179 of the DNA sequence that builds the sequel E enzyme, an adenine, the A nucleobase, has been accidentally substituted with a thymine, the T nucleobase.
Just one letter out of place.
Just one letter.
And because DNA is the instruction manual for building proteins, that one wrong letter changes the physical protein being built.
Exactly. This tiny genetic substitution occurs at codon 393. A codon is a specific sequence of 3 DNA nucleotides that tells the cell's machinery to insert one specific amino acid into the protein chain. Because the adenine swapped to a thymine, the instruction manual changes.
So what amino acid gets swapped?
The amino acid produced at position 393 changes from leucine, the L, to phenylalanine, the F.
So in the clinical literature, this is written as the L393F amino acid change. The sources also mention another frequently reported mutation nearby, the F397L substitution, which is a similar swap. What do these tiny singular amino acid swaps actually do to the physical squalene epoxidase enzyme?
Amino acids have different physical sizes and electrical charges. Phenylalanine has a large, bulky aromatic ring, whereas leucine does not. When you swap them, you change the three-dimensional physical shape, the folding architecture of the sequel E enzyme.
And you change it right at the critical binding pocket.
Exactly.
We can use a lock and key analogy here. The sequel E enzyme is a lock. Turbinafine is the master key that we engineered to slide perfectly into that lock, jam the mechanism, and stop the assembly line.
That is the perfect analogy. The L393F mutation physically alters the internal shape of the lock. The binding pocket becomes structurally restricted.
So the key doesn't fit.
Right. When the turbinafine molecule enters the fungal cell and tries to bind to the enzyme to shut it down, it simply cannot fit anymore. The bulky phenylalanine molecule blocks the drug from anchoring. The structural alteration physically reduces turbinafine's binding affinity to near zero.
But if the lock is mutated, how does the squalene fit? If the enzyme is deformed, shouldn't the enzyme be broken?
And that is the terrifying evolutionary genius of this mutation. The mutation warps the lock just enough to repel the artificial drug, but it completely, perfectly preserves the enzyme's natural biological function.
Yeah.
The natural squalene molecule can still slip into the pocket, bind, and be oxidized. The assembly line keeps moving. Squalene is converted to lanosterol, ergosterol is continuously produced, the cell membrane remains incredibly strong, and the fungus survives completely unharmed while swimming in a sea of turbinafine.
It's a molecular magic trick. And the clinical impact of this tiny genetic typo is quantified by a measurement called the minimum inhibitory concentration, or MIC.
Yes. The MIC is the critical metric in pharmacology. It is the lowest concentration of an antimicrobial drug that is required to successfully inhibit the visible growth of a microorganism in a lab setting.
So what is the MIC for normal ringworm?
For standard wild-type ringworm, the MIC of turbinafine is incredibly low fractions of a microgram. A standard pill easily achieves that concentration in the human skin.
But for these mutated T and indotiny strains?
The MIC values skyrocket exponentially, often requiring greater than 10 micrograms per milliliter of the drug to even slow the fungus down.
And we can't give patients that much drug, right?
Absolutely not. At those levels, standard turbinafine therapy is rendered utterly biologically useless. You cannot safely give a human being a dose high enough to reach a concentration of 10 micrograms per milliliter in their epidermal tissue without causing catastrophic systemic toxicity to the patient.
The drug has been entirely neutralized. So the obvious glaring question is, how did this happen? How did an ancient, slow-growing fungus suddenly evolve this incredibly specific, highly advantageous genomic mutation so quickly? Because evolution usually takes thousands of years.
This raises a profoundly uncomfortable question about human behavior and medical regulation.
It wasn't natural, was it?
The evolutionary pressure cooker that forged this super strain was not a natural phenomenon. It was largely driven by the catastrophic misuse of modern pharmaceuticals.
Specifically, the rampant availability and misuse of over-the-counter topical antifungal corticosteroid combination products. We did this to ourselves.
They really did.
Let me introduce an analogy here, because when I read how these combo creams work, it blew my mind. Applying a combination steroid and antifungal cream to a ringworm infection is the biological equivalent of trying to put out a fire in a building while simultaneously spraying the building's fire alarms with expanding foam so they can't ring.
That analogy perfectly captures the biological contradiction. Corticosteroids like hydrocortisone or betamethasone are powerful, localized immunosuppressants.
They shut down the alarms?
They act rapidly to reduce inflammation, restrict blood flow, and stop itching. From the patient's perspective, they rub the cream on the massive red plaque, the redness fades, the intense itching stops, and they feel incredibly relieved. They think they are cured.
But they haven't cured anything. They have just silenced the fire alarms.
Exactly. They have chemically deactivated the local cell-mediated immune response. The macrophages and neutrophils retreat. The skin's natural defense grid is completely taken offline.
So the immune system stops fighting the fungus. But the cream also has an antifungal in it, right? So shouldn't that kill the fungus while the immune system is napping?
In theory, yes. But in practice, these combination creams are often applied incorrectly. Patients apply them sporadically or at too low of a dose, or they stop applying them the moment the itching starts, which is far too early to eradicate the pathogen.
So we expose the fungus to a sublethal dose of the drug.
Exactly. You expose millions of multiplying fungal spores to a sublethal dose of an antifungal weapon inside a microenvironment where the host's natural immune system has been artificially paralyzed.
That sounds like a recipe for disaster.
It is the absolute perfect, unopposed environment for rapid evolutionary adaptation. The fungus multiplies rapidly. The millions of cellular divisions increase the chance of a random genetic typo. When the L393F mutation randomly occurs, that specific spore survives the weak antifungal cream while its neighbors die.
And then that mutated spore clones itself.
That single mutated spore clones itself, and suddenly you have selected for an entirely new, fully resistant superstrain.
We literally built an evolutionary training camp on our own skin, supplied it with weak weapons, turned off our immune defenses, and trained the fungus to beat our best drugs. And T-indotiny isn't the only nightmare emerging from this training camp. There is another variant detailed in the sources that has public health officials incredibly concerned. Genotype 7 or TMVAI.
TMVII represents an entirely new behavioral paradigm for dermatophytes. It is a highly distinct, newly emerging threat that has recently begun sweeping through Europe and the United States.
And how is it transmitted?
It is, for all intents and purposes, a sexually transmitted ringworm.
Sexually transmitted dermatophytosis,
Yeah.
a sexually transmitted skin fungus. That is a massive paradigm shift.
It is unprecedented. TMVII causes highly painful, persistent, highly inflammatory lesions that are specifically localized to the genitals, the pubic region, and the buttocks.
And it spreads differently than normal ringworm.
Rigorous epidemiological contact tracing indicates that, unlike standard ringworm, which might spread in a wrestling gym, TMVII is largely transmitted through direct, intimate sexual contact and the sharing of contaminated bedding between partners.
So the diagnostic landscape has just become a minefield. We have T-indotiny causing massive turbinafine-resistant plaques across the torso, and we have TMVII causing sexually transmitted genital lesions that mimic other STDs. This completely shatters the old playbook. How do clinicians fight an enemy that has out-evolved our frontline weapons?
It requires a massive forced paradigm shift in systemic therapy. Because terbinafine and the older drug griseofulvin are experiencing massive clinical failure rates against these superstrains, the new medical protocol mandates the use of a different class of drug.
What's the new frontline defense?
The frontline defense against these highly resistant strains has shifted to itraconazole.
But itraconazole is not a casual medication.
Yeah.
You don't just take it lightly.
No, itraconazole is a heavy-hitting pharmacological agent. It operates through a different mechanism, inhibiting the synthesis of ergosterol at a different stage. But the protocol to defeat these superstrains is brutal.
What is the dosage?
It requires a high dose, 100 milligrams taken twice daily, and it requires an extended duration of anywhere from 4 to 12 uninterrupted weeks.
And the major caveat with itraconazole is that it is heavily metabolized by the liver, right?
Yes. Because of its pharmacokinetics, maintaining a patient on high-dose itraconazole for up to 3 months requires careful ongoing clinical monitoring. Physicians must conduct regular blood tests to monitor liver enzymes and watch for hepatotoxicity chemical-induced liver damage.
Think about the weight of that. We are trading a relatively safe, short-course drug like terbinafine for a potentially toxic long-course drug that requires constant blood monitoring, simply because we forced the fungus to out-evolve us by overusing anti-itch creams.
It is a stark reminder that in the microscopic world, every action we take has an equal and opposite evolutionary reaction.
Let's take a deep breath and summarize this incredible, terrifying journey. We started millions of years ago in the dirt of the Mesozoic era, looking at a simple, soil-dwelling, keratin-eating fungus that evolved perfectly alongside early mammals like the Spinalestes. We explored the duality of its existence, the quiet, sexual telomorph mixing genes in the earth, and the aggressive, asexual anamorph cloning itself on the host.
We examine the sheer biochemical violence of the infection, the arthrochonidia attaching to the stratum corneum, germinating and secreting specialized keratinase enzymes to cleave the disulfide bonds of our skin, dissolving our structural armor into a liquid nutrient soup.
We mapped the human body's tinnias, from the sweaty, occluded interdigital spaces of tinea pedis to the dense keratin fortress of tinea unguium. We explored how zoonotic spillover from cattle, dogs, cats, and wild hedgehogs triggers massive, catastrophic inflammatory immune responses like carrions, simply because those pathogens lack the evolutionary camouflage to act as stealthy roommates.
We navigated the diagnostic labyrinth, breaking down the alkaline hydrolysis of the KOH prep, the physics of fluorescent pteridine metabolites under a Wood's lamp, the precise pH shifts of phenol red in fungal cultures, and the rapid barcode scanning precision of PCR DNA sequencing, all while recognizing the sobering statistical reality of a 15% clinical false negative rate.
We visualized the molecular machinery of terbinafine, acting as an immovable roadblock on the scoline epoxidase assembly line, starving the cell of ergosterol while simultaneously drowning it in a toxic buildup of raw scoline until it bursts. And we detailed the grueling 10-minute wet contact bleach protocols required to purge the resilient 18-month-old spores from our homes.
And finally, we confronted the biological monsters of our own making. The explosive global emergence of trichophyton indotinae and TMVI. We saw how human-driven genetic mutations, specifically the L393F point mutation that physically warps the binding lock of the sequelae enzyme, were forged in the evolutionary pressure cooker of misused steroid combination creams, resulting in unkillable, sexually transmitted superstrains.
Which leaves us with a profound and frankly deeply unsettling final thought for you to mull over. We have spent decades altering our immediate environment and self-medicating with powerful localized immunosuppressants and sublethal antifungals, all in a desperate attempt to eradicate what we ultimately viewed as minor, itchy inconveniences.
We sought absolute chemical sterility on the surface of our skin.
Exactly. But considering the explosive, unstoppable emergence of T-indotinae and the behavioral shift of TMVII, we have to ask a very difficult question. Are we accidentally curating an entirely new era of human microbiology? By relentlessly trying to chemically sterilize our stratum corneum, have we actively forced the rapid, unstoppable evolution of an entirely new class of unkillable, highly adapted anthropophilic symbionts? Fungi that are so perfectly mutated to resist our science that we will simply have to abandon our medications and learn to live with them forever.
It is a humbling thought. By declaring an unwinnable chemical war on a 125 million-year-old organism, we may have simply engineered our own permanent, indestructible biological roommates.
To everyone listening, thank you for joining us on this massive deep dive into the microscopic, rapidly evolving world of dermatophytosis. Keep questioning the world around you. Keep looking closely at the minor ailments we take for granted. And remember that sometimes the most profound and terrifying evolutionary leaps in history aren't happening in the jungle. They are happening right now, right on the surface of our own skin.